See this first question. A child came with complaints of multiple seizures, high fever, headache, neck rigidity, and the lab investigation of the CSF shows up what? Low glucose, high protein, and many polymorphs. So one thing is clear from this particular aspect only, that they are talking about which particular meningitis? Pyogenic meningitis. Fine. So polymorphs, low glucose, high protein, they all are features of what? They all are features of pyogenic meningitis. Then this particular word you see, pleomorphic gram-negative bacilli was seen in the CSF culture. What is the true statement about this causative organism which have done this pyogenic meningitis? The answer is C. Okay. It exhibits satellitism around the Staph aureus colonies or streak, you can say. Okay, and we are talking about which particular organism here? Haemophilus influenzae. Okay, and this was asked in the previous INI-CET exam. And what further it can be asked, in which other fashion it can be asked, that also we can discuss up. Okay, so first thing is this you should know that if these types of profiles is being up, we are talking about what? Pyogenic meningitis. Okay, important is bacteria जो हैं, these are like they love glucose. Okay, भुक्कड़ हैं वो glucose के, they love glucose, and that's why the CSF glucose remains very low, or it can become zero in them. So basically from the question, this particular hint, pleomorphic gram-negative bacilli, that was observed, and pleomorphic is which one, guys? Haemophilus influenzae. Okay, Haemophilus influenzae. Fine. Now you tell me Haemophilus influenzae. What is the other name of it? What is the scientific name of Haemophilus influenzae? That is P. first bacillus. Okay, everybody knows it. P. first bacillus is the scientific name of it. Is it capsulated or non-capsulated? So it is capsulated as well as non-capsulated. And the capsulated strains, they does more of a virulent infections like meningitis and epiglottitis. And the other ones, they does otitis media, the simple ones, sinusitis and all. So that you know. Fine. Now next time, what they can ask up, and first of all, let me tell you what is satellitism. Satellitism is, suppose you want to grow up, okay, you want to grow up Haemophilus influenzae on blood agar, okay, in between you have put the streak of Staph aureus, and you know Staph aureus is what? It is mainly beta hemolytic, okay. So what observed by growing up on the blood agar, this is blood agar, the Haemophilus influenzae colonies in this particular plate all around, they are small, but the colonies which are near to this streak, they are big in size and number. Okay, and why is it so? Because they have got the required factors X and V near to this streak, and that's why we say that they are looking like a satellite around the Staph aureus streak. This is termed as satellitism. Okay, is it fine? So ये होता है satellitism, and that is being shown on which media? Blood agar. Lekin Haemophilus influenzae loves to grow on what? It loves to grow on chocolate agar. Okay, it loves to grow on which media? Chocolate agar. Fine. Now, अब दूसरों का देखते हैं. Let's see अब the other options. अगर कहीं क्वेश्चन में बेसिट्रेसिन की बात हो रही है, if somebody is talking about बेसिट्रेसिन sensitivity and all, which particular organism you should look out for? So guys, you should look out for a gram-positive cocci, and that is obviously beta hemolytic strepto. Okay, so basically bacitracin sensitivity testing is being done to differentiate between what? Group A and group B strepto, if you remember of, and group B strepto and group A strepto. So group A is which one? Streptococcus pyogenes, and group B is which one? Streptococcus agalactia. Fine, so that you know. So which one is bacitracin sensitive? Group A is bacitracin sensitive, okay, and group B is bacitracin resistant. Okay, this is bacitracin resistant and this is bacitracin sensitive, and as such the antibiotic testing is done on which media, guys? It is done on MHA, but here you have to do it on blood agar, or you should put the blood into MHA. So this is an exception in that sort also. Fine, so that is an exception in that sort also. So if they want to ask up, in fact they have already asked in the NEET PG regarding the bacitracin resistant one, and they have asked up, this was one of the hints, and the question was regarding meningitis only. So obviously if they are talking about this, that is a bacitracin and all, immediately you should think for what? That is a beta hemolytic strepto. Fine. Then other ones, which produces non-lactose fermenting colonies on McConkey agar. Okay, so non-lactose or lactose, like that if the question is coming off, and suppose the question is telling about the lactose fermenters and all. So you know the lactose fermenter which causes neonatal meningitis. So guys, you know that is E. coli and Klebsiella. Okay, E. coli and Klebsiella, they are the ones which are lactose fermenters, and even they associate with meningitis. Which meningitis? Neonatal meningitis. Is it fine? So this also needs to inculcate that if it is asked in the form of lactose fermenters and non-lactose fermenters, then you have to think for these fellows regarding the meningitis, E. coli and Klebsiella. Now, talking about the alpha hemolysis on the sheep blood agar. So alpha hemolysis and all, if we talk about, then obviously it is not going towards that staph and all, because alpha hemolytic strepto is there, and that you know that is pneumonia, streptococcus pneumoniae. Then they are talking about what? Then they are talking about streptococcus pneumoniae, guys. That is pneumococcus, and this pneumococcus is the most common cause of meningitis in all age groups except the neonates, because in that it is streptococcus echinacea. Fine. So that also needs to be remembered, alpha hemolysis and all. So like that also the question will be coming off with these particular hints in the exam. Now let's see a particular table which, and you should tell up, okay, ki which particular organism we are talking about according to this particular one. See this particular question, that a gram-positive cocci in pairs and lanceolate shape. Tell in the comment section that what we are talking about, guys. Okay, tell in the comment section that what we are talking about. That is a gram-positive cocci in pairs, lanceolate shape. That is pneumococcus, guys. Okay, so like that with these particular hints also, the questions might come in your exam, and that is pneumo. So the pneumococcus, this might be their hint, and other hints which can be there in the question is, just now we have talked about, this is alpha, a proper spelling, alpha, A-L-P-H-A, alpha hemolytic. Alpha means what? A partial hemolysis, that is termed as alpha. Draughtsman shaped colonies on blood agar, Draughtsman shape, okay, that is carom coin appearance. And if you remember of that particular mnemonic which I used to tell up, bio, it is biopositive, okay. And what is biopositive? Bile soluble, inulin fermenter, optochin sensitive, okay. So these will be there in the exam, and they will be asking up which particular meningitis. So they are talking about streptococcal pneumonia, that is pneumococcal meningitis. Is it fine, guys? Now other one you see, the gram-positive cocci in short chains. Short chains. Chain, whenever you talk about chains, what we are talking about? Streptococcus. Fine. So beta hemolytic pinpoint colonies, remember pinpoint colonies are there with strepto on blood agar. And see this test, CAMP test. What is CAMP, guys? Christie Atkins Munch Peterson, okay, and it is resistant to the bacitracin. Just now we have talked about this. Can you tell me what we are talking about? Resistant to bacitracin, right? You are. We are talking about Streptococcus echinacea. Now talking about this, a gram-negative cocci in pairs, it is in pairs, intracellular, inside the pus cells also it is observed. It is oxidase positive and loves to grow on chocolate agar. Gram-negative cocci in pair, intracellular, inside the pus cells also it is observed. Oxidase positive, in fact catalase positive also, grows on the chocolate agar. That is also termed as modified chocolate agar, Thayer-Martin media. We are talking about what? We are talking about meningococci. Why gono? Why gono? Gono is meningitis. Okay. So gram-negative cocci, देखो मेनिंगो भी तो इंट्रासेलुलर होता है, मतलब it is both sides ना, it is extra as well as intra, and it is oxidase positive, it grows on chocolate agar. We are talking about meningo, not the gono, okay, because here we are talking about meningitis, and meningitis obviously is done by what? Meningo one. Pleomorphic gram-negative bacilli. देखो आ गया है भी. This particular hint was there na. Pleomorphic gram-negative bacilli. Satellitism is observed in blood agar with the staph aureus streak. Okay, and this particular X and V factors, because this particular organism loves to have these X and V, and it shows up this phenomena satellitism on the blood agar. So obviously what we are talking about? We are talking about Haemophilus influenzae. And in the above one we are talking about what? We are talking about meningo. Okay, we are talking about meningo. Now this one, a gram-negative bacilli, and the identification is based on colony grown on McConkey agar and the biochemical reactions. So McConkey agar have come into scene and gram-negative bacilli. So what we are talking about, along with the biochemical reactions like IMVIC and all, and IMVIC I am telling you that is plus plus minus minus. So which organism am I talking about? So I am talking about E. coli, guys. Okay, I am talking about E. coli, and the same thing if I want to change up to Klebsiella, I will do what? IMVIC minus minus plus plus. Okay, minus minus plus plus. Is it fine? Now further, the Indian ink stain. If this particular name comes up, Indian ink stain showing budding E cells with refractile capsule. Okay, and the latex agglutination test is also coming positive for this capsule antigen in CSF, and this also can grow on blood agar or chocolate agar or SDA, showing mucoid white colonies. We are talking about which fungus? We are talking about the Cryptococcus, guys. We are talking about what? Cryptococcus neoformans, Indian ink staining. Now telling about this, the detection of viral nucleic acid, DNA or RNA in CSF by PCR or real-time PCR. This is the story of all the viral meningitis, and for all of them we are going for what? PCR. That is the real-time PCR. Okay. Now this is obvious, the ZN stain of CSF showing the acid-fast bacilli, detection of specific genes in the CSF by PCR or gene expert, and the growth is also being observed where, guys? On the Lowenstein-Jensen media, rough, tough and buff, and down it is written up, mycobacterial growth indicator tube. So which organism we are talking about? So we are talking about Mycobacterium tuberculosis. Okay, what we are talking about? We are talking about the Mycobacterium tuberculosis causing up TBM, that is TB meningitis. Is it fine, guys? So these are the ways by which the question can be asked up by giving up the cultural characteristic of that, and indirectly they can ask up regarding the pyogenic meningitis. And another important point which I like to tell up is, it is not necessary that in the culture you could able to find out that organism. In around 60% cases, the culture yields negative result. In 40% only the culture comes positive. This also was there in one of the exams, that culture is coming negative, but all those features, they are of pyogenic meningitis. So which particular one? So everybody got confused, culture is not showing anything, so it is not bacteria and all. So there is a clear-cut line in Harrison that mostly times the organism is not being cultured. So this is the way to approach up these types of questions, and if you know these particular properties of them, it's a cakewalk for you. Okay.